We're probably going to get started here pretty soon, but before we do, I wanted to let everyone know who attended last week's meeting that I am still editing the video, but should be able to get the link sent out this afternoon along with the survey information. So please hold tight on that. Now that I've got the process figured out, it shouldn't be too much longer, and this week should go much smoother.
Heather, it looks like there's some people in the waiting room. Do I need to admit them, or because it looks like I have host capabilities?
I've got them. I've been
Okay.
admitting. Yeah, you're good.
Heather, this is Jake. I'm just waiting on your signal to go.
Okay. Okay. Well, it's 3 after, and we've got 43 people in here right now. So I'll keep letting them in as they come in, but I would say go ahead and go whenever you're ready.
Okay. Great. Yeah, I hope everybody can hear me. My name is Jake Michaelson. I'm an associate professor in the Department of Psychiatry here at the University of Iowa. I do a lot of my research is computational in nature, and so I was asked to give a lecture today on bioinformatics. And I hope that this will be, I have not given this specific talk before. I made some new slides for it, and it's going to be pretty practical, I would say. It's going to be, because the questions that I typically get about bioinformatics are less of the nature, tell me more about the latest advances in bioinformatics. I rarely get questions like that. What I do get questions about frequently related to bioinformatics is how can I get started in bioinformatics? What is bioinformatics? How is it different from, say, biostatistics or computational biology? And what are the career prospects in bioinformatics like? And so I'm gonna talk a little bit about the science, but I'm gonna spend more time talking about these kinds of practical issues and hopefully addressing these practical questions. So I've made a couple of assumptions about the audience today, and I wish I could be with you and see you and interact with you a little bit more than we're able to over Zoom. But one of the assumptions I've made is that, and I hope this is fairly correct, and that is that you are earlier on in your training or in your career, and secondly, that you are at least a little bit curious about what bioinformatics is. I've probably also implicitly made the assumption that maybe many of you are not currently practicing bioinformaticians. So I hope that those assumptions are largely correct. And I realize there are going to be some exceptions within the crowd that we've got together today. The other thing I want to say is that I hope that, I don't know what the typical format is like or what it allows, but I hope that there will be spontaneous questions. You do not, as far as I'm concerned, you do not need to wait until the end of the talk to ask questions. And so I hope that you will feel at liberty to either ask a question through the chat, or if it works better, to unmute yourself and ask a question. So I hope that you don't feel inhibited in that regard. Now, before I dive into the talk, I wanted to start with a quote from Adele Cutler, who is a statistician and a machine learning researcher. And she was one of my earliest mentors when I was an undergraduate student. And she came up with a very widely used regression and classification method called random forests. And she said something about random forests. It gained a lot of popularity, and a lot of people began to rely on the predictions that were made by this approach in sort of an unquestioning way. And so in response to that, she wrote the, I think I'm going to go ahead here and just run through the list and make sure people are muted. There we go. Okay. So she said that random forest is an example of a tool that's useful in doing analyses of scientific data, but the cleverest algorithms are no substitute for human intelligence and knowledge of the data in the problem. Take the output of random forest not as absolute truth, but as smart computer-generated guesses that may be helpful in leading to a deeper understanding of the problem. And so I'd like that, I wanted to begin with that because I don't want people to come away with this operating under the assumption that computers are going to solve all our problems and computers are going to help us understand everything there is to know about biology. Really, it's a partnership with computers that is the essence of bioinformatics, and that's why I put up this this illustration here of a human and a robot working together, and that's sort of illustrative of what bioinformatics is all about. We want to use computers to do things that the human brain is not particularly good at, and that is performing repetitive computations on extremely large volumes of data. But we want to marry that with the underlying human intellect and with that expertise and the knowledge of the problem. And when you put those two together in a complementary way, then that is when you arrive at discoveries. And that's when bioinformatics is at its best. So the rough outline of what I'm going to talk about today is, first, I'm going to, probably the first half or so, probably first half of the talk is just going to be around the question of what is bioinformatics. And then one section that I thought was going to end up being longer, but I really did not put very much into, was how has, mostly because I realized in putting it together, I was like, this is like a whole semester course, not something that I can put into a few slides. So I just put a few slides together on how bioinformatics has changed biology and medicine. And then finally, I'm going to finish with some real world examples of bioinformatics as applied to psychiatry and neuroscience. And these are examples out of my own group. So for this part of the talk, I'm gonna introduce my story briefly and how I got into using computers in biological research. And then I'm going to talk a little bit about who bioinformaticians' nearest neighbors are, and that'll make sense in just a minute. And then I'm going to spend some time on talking about how to get started in bioinformatics, and then a little bit about formal training in bioinformatics, careers in bioinformatics, and then collaborating as a bioinformatician or other kind of computational person. So I went to an agricultural university. Utah State University is an ag school. And I was majoring in biological engineering, and very early on in my undergraduate career, I happened to attend a talk of a visiting researcher. And because I was in an ag school, what he was talking about was, he gave a talk on genes in cattle that influence the marbling of their beef. Now, if you don't know what marbling is, I've included a picture here, and it's basically the degree to which fat is kind of interdigitated into the meat of an animal. And it's much more desirable to have high marbling because that gives you a moister, more juicy cut of beef. And so that's a very desirable trait. And if they can figure out the genetic basis for that, well, then obviously they'd like to turn that up quite a lot in the cows that, or the cattle that they raise. And so he was, and you can see down below, this is an example. This isn't a particular one that, because it was much older than this paper actually. But there are specific places in the cow genome where, in the bovine genome, where that controlled this attribute of marbling. And this was the first, I was very young and very inexperienced, but this was like my first interaction or first exposure to this idea that genes and changes in genes have a pretty pronounced effect on phenotypes or on physical traits. And even more than that, it was the idea of prediction that fascinated me most about using biology or using computers in biology. So this idea that you could predict, in this case, it was predicting what a cow's beef was going to be like based on their genetic profile. That was something that I found incredibly fascinating. Now, the issue was that at the time, this was in the early 2000s, there really was, although, you know, you could make the argument that bioinformatics was definitely around, it had not really penetrated, it certainly hadn't penetrated this agricultural university where I was as like a field. It was not something that you can major in, although today you can definitely, there are many bioinformatics degree programs, but at the time it was not a degree program. And so I did a 5-year bachelor's and master's program at Utah State University in biological engineering. And then I decided that I wanted to go overseas to do my PhD. And so I did a PhD in computational systems biology in Germany at a university called TU Dresden. And it's kind of an engineering school. And in fact, I've got a picture of it right there, that lime green floored building with those odd art installations. That is the computer science building at TU Dresden. And that is where I defended my dissertation. After that, I did a postdoc with Jonathan Savat at the University of California, San Diego in psychiatric genetics. And since 2013, I've been on the faculty in the Department of Psychiatry at the University of Iowa. And the general theme of my research is to understand the brain and to understand its strengths and its liabilities using genetics, because of course genetics provides the blueprint for how a brain is patterned, how it's developed, how it ultimately wires itself up together. Obviously, experience and environment are a very important part of that, but the brain kind of lays, or the genome lays the foundation or lays the blueprint for that brain. So we study things like language ability, like autism, sexual and gender identity, and also the relationship between creativity and mental illness, because there's a very fascinating relationship there. So that's kind of, in a very quick summary, my relationship with computing and biology. Now, even though I've been in bioinformatics for a while, or I've done bioinformatics work for a long time, I thought, you know, I know how I would define it, but I'm not sure how other people would necessarily define it. And so I resorted, which I don't like doing, but I resorted to looking up on Wikipedia how they define bioinformatics. And this is what they said. Bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data, in particular when the data sets are large and complex. And I actually think that's a pretty good definition. That's pretty congruent with my experience. It is definitely very interdisciplinary, meaning that people kind of arrive at bioinformatics from lots of different directions. Some come from biology, many come from computer science or statistics, and they meet in the middle. The other thing is that bioinformatics tends to be pretty methods oriented, and I'll talk more about that in just a second. And then the other requirement is that for something really to be considered bioinformatics, the data that you have to be processing on needs to be large and it needs to be complex. Now, this is something that I think is hard to define objectively, but this has been my experience in that bioinformaticians have more, or they tend to have more of an engineering mindset than a scientist mindset. And what I mean by that is that their focus tends to be on solving a particular problem and on the tool that does that. And while they are very important in enabling discovery in science, they're rarely the ones asking the scientific questions. Usually it's another kind of a scientist who has specific questions that they're investigating, and then they employ a bioinformatician to help them understand the data and help them to do things with the data that they are not able to do themselves. The other thing is, it might just be me, but I often hear he's a bioinformatician or she's a bioinformatician more than I hear I'm a bioinformatician. And what I mean by that is that it's not something that I find is really often used by people who do this kind of work. It's more often applied as kind of an imprecise and kind of a hand-wavy label that's applied by scientists who are trained in other domains, but applied to people who help them with their data challenges. Let's see, I think I can see a few things in the chat here. Okay, oh, it's just audio issues. Well, I hope people get those sorted out. Okay, so there's that. So now I want to talk just for a moment about what bioinformatics' nearest neighbors are, because you might hear people kind of use these different words interchangeably, but there are subtle differences. Although there are definitely commonalities between them, there are subtle differences between what the emphasis is and what they like to focus on. So in computational biology, a computational biologist is also good at computing, just like a bioinformatician would be. But generally, they're more interested in the biological insights, right? So they're more interested in discovery and learning about biology than they are necessarily in the tools that help them to make those discoveries. So this is generally a PhD. This would be kind of most computational biologists that I've encountered are faculty. There aren't as many kind of technician level positions in computational biology, simply because of that fact that they're more interested in like asking the questions. Now with biomedical informatics, that's very close in word space to bioinformatics, but it's actually a pretty different emphasis. There's a specific and a strong emphasis on research using electronic health records. These are often housed within a hospital, for instance, biomedical informaticians. There's a strong emphasis on databases and a strong emphasis on natural language processing and text mining. The folks that I've encountered that do this are either at the master's or PhD level typically. And then there are biostatisticians. So like bioinformaticians, they're often part of a large interdisciplinary team. But a key difference is that for a biostatistician, the data need not be large to require a biostatistician. So you may have You may be working in a clinical trial that has data that's very tractable. It doesn't have a ton of variables. It doesn't have, maybe it doesn't even have a ton of participants, but you need a statistician who has a certain degree of domain experience to be able to answer specific questions about whether the numbers are sufficient and so forth. These are often master's or PhD levels, and then the PhD level biostatisticians are generally more interested in developing the math and the theory behind a new approach than they are in developing and maintaining software and pipelines. So I hope that that illuminates or clarifies a little bit the distinctions between bioinformatics and then these other related fields that combine some kind of computing or some kind of numerical aspect with biology. So now I'm going to turn to how to get started in terms of informal training, because this again is a question that I get asked a lot. So you're going to need a few different ingredients. You're going to need some basic computing skills. You're going to need some kind of a large data set, because again, that's kind of part of the definition of bioinformatics. If it's not a large data set, you might be doing potentially biostatistics or something else. But you also need a question that the data can shed light on. And you need some computing resources that are matched to the data. And in addition to all that, you need some kind of a guide and probably all of these. So that could be tutorials or scholarly papers that you see. It could be an online forum, or it could be an actual human being. It could be a mentor. In terms of the basic computing skills, before we even get to like the programming and stuff like that, this is my personal opinion, having done these things myself, but it is incredibly informative to have the experience of building a computer. And if you don't have the means or the access to the materials to build a computer, see if you can find an old one that you can take apart. Because since a computer is such a vital tool in bioinformatics, it's really important that you understand what all the different internal parts do, because each of those internal parts is going to impose a different kind of a limitation on the kinds of problems that you are able to address with your bioinformatics skills. So it's really important to understand how a computer works and what the different parts are. So for a less expensive option, you could buy a used computer. I know here at the university, they have surplus stores where they sell off old computers that they're retiring for a fraction of the original price. Or you could buy a Raspberry Pi, which is a small single board computer that, depending on the specifications, goes from anywhere about $30 to $60. Or you could go the more thorough and useful option, which is to build a system yourself and to buy the individual components, which is kind of what I did when I first was starting out. And there's this great website called ppartpicker.com that is an incredible resource for anybody who wants to build a computer. There are a lot of tutorials there. There are literally thousands of documented builds that people have made for different purposes. So if you want to build a gaming PC versus a PC for deep learning versus, you know, a PC for a database or something else, there are lots of different guides and walkthroughs that will help you through that process. So I think that's vital. The next is to get away from the operating system that was just on the machine when you bought it. So very often that is Windows, or if you buy a Mac, that's the Mac operating system. And installing Linux on that computer, which is a sort of Unix-like operating system, and Ubuntu is a popular and a well-supported choice for Linux. But as you install that, you again become much more familiar with how a computer works, and you're a little bit closer to the processes that are going on, and you'll be in a better position to understand the limitations of the things that you're asking a computer to do. And so part of installing and working with that Linux operating system is to work with the terminal, which is basically just a screen that has a text prompt in it, and you type in commands. Let's see, I see Abigail is asking, by building a computer, do you mean physically putting the hardware together? And yes, that's a good question. Yes, that's exactly what I mean, is physically putting the hardware together. Because again, there's no substitute for understanding the layout, understanding what a motherboard is, where a processor goes, what the cooling systems are that are in place to remove heat, to understand where memory goes, to understand how different parts can fail. And all of that just kind of starts, that contributes to your intuition for how a computer works. And again, what's important are the constraints, which I'll talk about in just a few slides. So yeah, back to Linux. You install Linux on your computer, and you start to use these little funny named pieces of software like SSH that allows you to log in remotely to a computer and run commands as if you were actually physically present at that computer. To shell scripting and other programs like sed and awk that allow you to manipulate files and manipulate text, especially. The other part that's kind of a rite of passage is learning how to compile a program from source code. So a lot of times the tools that you'll use as a bioinformatician are not things that you can just download from the Apple software store, or from, they're not ready to go pieces of software. A lot of times you have to compile them from the source code in order to get them to run on your system. And then there are other useful things. This is not an exhaustive list, but things like regular expressions, which is a system for matching text. Okay, got a question. Would disassembling other devices that are not laptop or desktop computers, like a calculator, small radio, etc., help too? I mean, that doesn't hurt, but those are not tools that you're going to be using routinely in bioinformatics. But you would be surprised at how much they have in common. In terms of, they've got power supplies, they've got circuits, they've got resistors and capacitors and things like that. So that, I think, helps with general curiosity, but it is more useful to actually do that with a computer. So a lot of the things I'm going to talk about in the rest of the talk are going to presuppose a knowledge of R. Python is also a great language to learn, and in many respects, it's more useful than R simply because it's a general purpose programming language. So if you wanted to learn a programming language and you weren't sure whether you were going to continue on in bioinformatics, Python would still help you a lot if you were still interested in computing, whereas R is very specific to analysis of data. So if you're never going to do data analysis again, then R would probably be of limited utility. But if you're very interested in data analysis, my opinion is that R is actually superior to Python for that particular purpose. So there are some resources there that I've listed that I hope you'll look into. I also talked to my grad students and got their recommendations on things that help them getting self-started. One of the things that was recommended was StatQuest, which is a YouTube channel by Josh Starmer, and the URL is right there. He's got really great bite-sized vignettes on different aspects of statistics and even machine learning. And he even has a whole series on high-throughput sequencing, which you may find of interest. Then there's also learning how to visualize data with R, and there's some basic and advanced resources there. And then the Bioconductor website. Bioconductor is a collection of packages that work, that are part of the R ecosystem. So they're R packages. And they are specifically for bioinformatics. Just realized I spelled Bioconductor wrong there. I might just fix that spelling. Oops. So nobody gets too confused. Bioconductor, there we go. So they've got a lot of great resources on the Bioconductor website. So if you go to the Learn section, and especially the common workflows, that will give you a lot of really detailed, but also very user-friendly documentation on how to start off with bioinformatics targeted at specific areas of biology. So the next step is to find some data. And the fortunate thing is that most tutorials are built around a readily available data set. So if you find a good tutorial that is sort of on a topic that you're interested in, almost all the time, they will have a data set that you can easily access and that you can play around with. But that might not be enough for you. Once you've kind of mastered that tutorial, you may be interested in exploring other kinds of data. And so as you read papers, if you really want to quickly arrive at whether there is data associated with the paper or how you can find it, just do a Control F or find, search through the text of the article, and look for words like available, because usually they'll say the data are available at, or can be downloaded from, or the data have been deposited and are accessible with this accession number, right? Or you can look in the supplementary data as well. There are also some really good high quality public data repositories, and I certainly cannot enumerate all of them right there, but probably the best one to get started with would be if you're really interested in bioinformatics, is the Gene Expression Omnibus. This is a collection of thousands and thousands of experimental results that were obtained either using gene expression microarrays or other technologies like RNA-seq. And it's very searchable. You'll be able to find data sets that are on a topic of interest, and you'll be able to reanalyze them. You'll be able to see if you can reproduce the results that were presented in the paper. And I think this is just a fantastic resource for getting data that you are interested in. The other thing I'll say is that the journal Nucleic Acids Research, which is a pretty good journal in bioinformatics, especially in sequence-based bioinformatics, they have an annual database issue, which is a tremendous resource for getting an overview of how things have evolved in the last year and what new data sources are out there and what big databases have had substantial updates in the past year. So that's, I would highly recommend that you look into that if you're looking for good sources of data.
Jake, you've got a question. Jake,
yes.
You've got a question over here in chat. It says, what should I learn first, Python or R?
You know, different people tell you different things, but for my money, if you don't have any programming experience, people generally find R a little bit easier to start off with, or at least it's easier to immediately get to kind of a minimal level of competency. So I would suggest R, and I think, you know, the built-in visualization tools tend to be a little bit better and easier to arrive at publication quality graphics. There's a really good, very solid community around bioinformatics with R. Most bioinformaticians that you'll talk to probably use R first and foremost. Many of them will also know Python. For instance, if you get into deep learning, Python is definitely the dominant language in deep learning, but that's kind of a separate issue. Okay, so the next part is, if you're doing this on your own, it's really important to have a question, because you can't just aimlessly analyze. You know, I guess you could just visualize certain aspects of the data. Maybe that's interesting in and of itself, but you need something that will drive you and kind of continue to propel you through the parts where you get stuck. So one way that you can develop a question is when you're reading papers, you can ask yourself, what questions were left unanswered by this paper? So one tip that I would say again is to use Control F and look for words like unclear or unknown or unresolved. And usually those are markers of the authors articulating what the limitations of their work is and what is still unknown in the field. And also to focus on the conclusion. That's usually where these things pop up. Don't spend time on questions that you find uninteresting, at least during your training phase. So it may be that there's a tutorial and it seems like a pretty good tutorial, but ultimately the biological question that they're kind of using and picking apart in the tutorial is not very interesting to you. What I would suggest is that you take the basic parts of that tutorial and apply it to a question that you're much more interested in, because it's going to be your passion that will push you through the parts where you get stuck. And if you don't have a passion for the question, you're just going to stay stuck because you're not really interested in the answer. Now, kind of as you mature and as you go through your career and advance to a later stage, you need to develop the ability to see the interesting part in any question. And this will make you a better collaborator. So for early stage, try to focus on questions that you are interested in so that you can have that motivation, but recognize that as you mature in this field that you need to be able to see the interesting parts in anything. Now, in terms of computing resources, there are lots of different sizes of problems. Some problems in bioinformatics you could do on a Raspberry Pi, which I've pictured right there at the right. Others would require access to a supercomputer like the one pictured below. In my experience, most routine problems in bioinformatics can probably be done on a decent laptop, and if not that, on a desktop computer or workstation. If you are working with larger data sets, meaning there are more individuals in the data or more features or variables or attributes, that is going to require more memory. So again, this kind of goes back to understanding the different parts of a computer, and building a computer can help give you an appreciation for this. You're going to need more memory to hold all that data as you manipulate it and perform transformations on it. If you're doing a lot of simulations or permutations. Is there a question? I'm having a hard time telling if that's a question or if it's just somebody unmuted.
She just got into the room, so I'm assuming she was unmuted.
Oh, okay. All right, then we'll go on. So yeah, if you're doing a problem, if you're working on a problem that, oops. Oh, geez. If you're working on a problem that has a lot of iterations or permutations, that is something where you want a really fast processor, or you want more processing cores. And if you're working on problems that involve a lot of matrix algebra and vector operations, a lot of times those can be accelerated substantially with the GPU, which is a graphics processing unit. Okay, in terms of finding guidance, so look on, again, this is kind of referring back to the bioconductor workflows, and there's also a video channel that they have where they have videos that will walk you through these things. With papers, a good exercise to do is, given the data and some sample code, which the best papers will include both of those, they'll make access to the data freely available. And they'll provide the code that they use to analyze it, and just see if you can replicate what they found in the paper. And a tip here is, if you run into trouble, before you email the authors, and you should always email the first author, because usually the last author, even though their email is the one that's on there and they're listed as the corresponding author, they very rarely are going to be able to answer your question. It's the first author who did the work and is going to be able to point you through those things. You should probably CC the corresponding author, but the first author is the person that's going to give you the answers. But before you even do that, though, see if there are more appropriate channels for help. Like, is this a software tool that has a mailing list, or is it a software tool that has a GitHub page, or some other means of support? Try going through those channels before you resort to emailing the author. Now, obviously, I'm sure most of you are familiar, or at least have heard of, websites like Stack Overflow and its cousins like BioStars. Those are online help forums for a variety of things, and BioStars is specific to bioinformatics, and many questions have already been answered quite well on there, so it's good to see if your question's already been answered. And then websites like Reddit have a bioinformatics subreddit. There's a really good machine learning one. It's not specific to bioinformatics, but it's a great source of papers and recent developments. And then there's no substitute for having a human mentor, somebody that you know or that you can know. And my advice there is to look for your T+1. And what I mean by that is, if you are currently an undergraduate student, the person you should look for is a graduate student you might be able to work with and that you might be able to help. I know in my lab, typically grad students have projects that they don't really have time to work on themselves, but are interested in. And if somebody came along and had the know-how and had the time to do it, they would be glad to mentor that person. If you're a grad student, see if you can find a postdoc. and so forth. And finally, in terms of the informal training, you should try, once you've done something, you should try to find an outlet for what you've done in terms of sharing it. So a GitHub page is probably your best option. And the reason for that is that for your career options, this is becoming more and more mandatory because it demonstrates that you prioritize reproducible science, that you prioritize good code quality because people can see the code that you've written, that you also are willing to take contributions and corrections from the community. It demonstrates all those things. And if not a GitHub page, then being active on social media or on a personal website, you can make a blog post about your experience in analyzing this data. I've given some examples there of some really good instances of communicating that sort of thing. Now, that's all informal training, okay? And informal training is great, and bioinformatics is great because it lends itself really well to informal training. There are a lot of resources out there for informal training. And so being successful in bioinformatics will require both, in my opinion, both the informal training, because that demonstrates that you have the raw talent, that you have passion, that you have the ability, and that you have the drive to do things on your own. That's all really important. What formal training provides, though, that I don't think that informal training really can, is that it fills out your blind spots, right? So if you were completely self-taught, you would naturally just kind of only gravitate towards problems that you find interesting or methods that you find interesting, and you would probably end up with a lot of blind spots. And what I mean by that are areas of research or areas in the field or technical details that you need to be aware of caveats and things like that, that you can only really get when you participate in formal training. The other thing that formal training does is it facilitates connections and it provides opportunities because it puts you in that environment where people are kind of networked together. So as you're considering the question of where to get formal training, again, this kind of goes back to what I said before of imagining where you want to be in 5 years, and then think about who is at that point. And you should try to interview people who are at that point. So maybe it's a graduate student in a particular lab, and you should get in contact with one of those graduate students and ask them how they got there, what they thought was vital in them getting to that point, and maybe what they, well, not just maybe, but definitely what they would have done differently, because there will be things that they would have done differently. Now, regardless of where you go, because nowadays we're fortunate that many institutions have a curriculum that will support development in bioinformatics. These are generally the kinds of courses that you should strongly consider taking. So for math, definitely linear algebra, because that teaches you about matrix and vector operations, which are kind of the foundation of things like principal components analysis and distance measures and clustering and things like that. Also, graph theory is very strongly rooted in linear algebra. Statistics, very important. I would say arguably more important than a strong math background is a background in statistics. So classes on linear regression, on probability, on design and analysis of experiments, and multivariate analysis are all key ingredients for bioinformatics education. In terms of computer science, there's you need to get some basic programming skills, kind of this on the same level that I was talking about before of your self-guided stuff. Databases, web development, mostly for dissemination, because maybe you make a database and you want to have some kind of a way for people to access it. And so you need to know how to design a simple website that can provide access to database search results. And then machine learning and deep learning are also an important part of that. And then in terms of biology, molecular biology and biochemistry are important for your foundations, as well as cell biology and definitely genetics. But one of the most important things that people might gloss over in terms of the formal training is looking for these more formal internships. So you should ask your professors if you can work for them. And most professors are doing research. And if you're lucky, they'll take you on in the first place. And if you're even luckier, you might get paid depending on what you bring to the table. But remember that you're in this for the experience and for the reference letter, potentially for graduate school or for a career. And if you don't get a response and you really want to work in a specific group, make sure to connect with the graduate student in the lab, because the professors are typically very busy and get a lot of email. And so you might just not get a response, where you might get more of a response from a grad student or a postdoc who's in the lab. And you should stay, if you determine that you like that position, you should stay there for as long as you can, because a solid stretch of experience is much better than multiple short-lived internships. And then those internships can lead to connections for grad school and beyond. In terms of careers in bioinformatics, there's the obvious faculty, but very few make it to that point. It's not very common to see somebody's title as professor of bioinformatics, although that there definitely are cases of that, but usually there's that person has expertise in something more specific or more applied. You know, for like, you know, I'm an associate professor of psychiatry. I'm not a psychiatrist. I do a lot of computational and genetic research in psychiatry, but I'm not a professor of bioinformatics. And then there's obviously, you can be a staff scientist in academia or in industry or work for a government lab. And the last thing I'll say about careers in bioinformatics is that over the past 20 or so years, bioinformaticians started off as being impossible to find, meaning that the job market was very favorable. And then it proceeded to hard to find. And then currently, I would say that it's hard to find a good one. I've heard a lot of people say that. It's not hard to find somebody who has bioinformatics on their resume. What is still hard to find, though, is somebody who's quite good. So the labor market for employees is pretty good, but your challenge is to differentiate yourself from lower quality applicants. So the obvious way to do that is to have first author publications, to have a recommendation that basically says, I don't want this person to go because they're so valuable. And then evidence of activity in a community, like having a productive GitHub page, and evidence of your communication skills. So collaboration is really important in bioinformatics. It demands collaboration, actually. And the irony is that it demands collaboration, but most of the people who are drawn to bioinformatics or have those inherent skills are introverts, which is sort of a sad reality. And so the ability to speak another scientist's language is critical. And you'll find that you'll often have to restate the problem and goals as you understand them to make sure you're both on the same page. And it's important to be involved early on in the process so that you're not just doing a postmortem on another person's data. Okay, I am taking way longer than I thought it would, so I'm going to skip over this and just say, yes, it has changed biology and medicine quite substantially. I realize that's not a very satisfying answer, but I'm going to skip over that because I want to get to a couple of examples from my own lab of bioinformatics before we end. So here's a picture of my lab when we were decorating Christmas cookies, and I think that was about a year and a half ago. I'm going to tell you about a few different projects coming out of my lab. So the first one is cerebroViz. This was published in Bioinformatics, the journal Bioinformatics, and the problem was how to visualize gene expression in an anatomical context, because that sounds like a fairly simple thing to do, but there was no tool to do that. We do a lot of work with the brain, we do a lot of work with gene expression, and we wanted to be able to visualize it as it is in the brain. And so we developed an R package that allows you to kind of easily paint gene expression values onto an anatomical image of the brain, and to do that, importantly, programmatically. So you're not in there with the mouse and pointing and clicking, and it's all programmatic. And that uses SVGs, which are a kind of XML document. The next one is drug repositioning. So here, the problem was that medicines for epilepsy don't work for everyone. Sorry about that. I'm not sure. Zoom just muted me for a second. I hope that wasn't very long. So the problem here is that medicines for epilepsy don't work for everyone, and there are some people who have what is called treatment refractory epilepsy, and we want to be able to find therapies that will work for them. And so the solution here is to discover epileptic signatures of gene expression. Get out my pointer here. So here we took brain tissue samples from people with epilepsy, and we looked at the tissue that was seizing or had the seizures and compared that with their tissue that was not having the seizures. And then we did RNA sequencing and identified how genes were differentially expressed. And then we use this database called the connectivity map, which basically tells you, it's a database of a whole bunch of different drugs applied to different kinds of cell cultures. And then they looked at gene expression to see how gene expression was perturbed. And so what we did is we looked for drugs within this database that reversed the patterns that we saw here. And then we tested those. We had some candidate therapeutic compounds, and we tested them in a zebrafish model of seizure to see which of those might actually be effective. And it turns out that there was a huge enrichment of efficacy among the drugs that we identified as potential new candidates. So yeah, that was published a little over a year ago. This next problem is called, the tool that we developed called TSAN. And the problem is that in general, we have no idea what non-coding variants do. And specifically, we don't know which tissue non-coding variants might be most relevant in. This is a problem in brain research because we know non-coding variation is very important in influencing brain phenotypes, but we just don't know where to begin with non-coding variation. So here we integrated a variety of genome scale data sources, which are shown in this schematic here. And we wanted to predict which variants were going to have an effect and in what tissue type. So in this paper that was published in Bioinformatics in 2018, we showed that this score cleans up GWAS signal quite substantially and improves the calculation of polygenic risk scores as well. And that basically happens by just focusing on the tissue where these variants are actually having an effect on the phenotype. So that's TSAN. More recently, we published a method called forecASD. And the problem there was that autism gene discovery, autism is primarily a genetic condition. So over 80% of the variability in autism diagnosis has a genetic component to it. And so gene discovery is really slow and it's really expensive and it requires a huge number of people. And so the solution that we proposed here is to integrate genome scale data to learn what constitutes an autism risk gene. So in other words, in terms of its brain gene expression profile, when is it expressed in the brain? Where is it expressed in the brain? In terms of gene networks, where do autism genes tend to reside in those gene networks? So forecASD is a method that provides a single score that forecasts, and that's what the play on word is. By the way, if you're in bioinformatics, you have to come up with these little acronym, funny little names for your methods. It's sort of like an unspoken rule. So forecASD forecasts what the as yet unknown autism genes are, and that they will likely be uncovered in future genetic studies. So the cool thing about this is we developed this several years ago, and there have been a couple of major autism genetic studies that have come out that where our predictions that we made back in the day have borne out, and that as new autism genes have arrived on the scene, the predictions made by forecASD have been vindicated. So here's an example of predicting activated neurons. This is single cell RNA sequencing. And we're using just the transcriptional profiles of individual cells. Here you can kind of see them clustered out to predict which of these neurons were recently activated. Now this has strong implications for learning and memory and identifying cells and neurons specifically that are involved in certain kinds of memories. So FOS is a gene that is well known in the literature to mark cells that have recently been activated. But the problem is in single cell gene expression data that there's a lot of sparsity. In other words, FOS expression, even though it's expressed in cells, it's not recorded there because there's what's called dropout, and it's just not observed in every cell, even though it's really there. It's just kind of a technical limitation. And so what we did is we took all of the rest of the genes in the transcriptome, and we used a deep neural network to predict the expression of genes that we know are markers of this cell activity. We have a bottleneck here in the neural network. And the activity at this bottleneck is strongly correlated with the expression of all these genes. And the activity at this bottleneck is basically a proxy for the activation state of a neuron. And so over here in the right-hand panel, you can see that we have a much better idea of which cells are actually active based on the whole transcriptome rather than a single gene. So that's another application of deep learning in our lab. I think this is the last one. So we were also very interested in brain imaging. And so the problem here is that brain imaging segmentation, or the regions, are largely defined anatomically, and that may miss some important phenotypic associations. And so our solution is to use deep learning to infer the structures that are linked to phenotypes. So in other words, we're trying to get, to have a neural network tell us what the structures are, and then those structures might not actually conform to classically defined anatomical structures. So we remove that constraint. And so in this example, we were using 3D convolutional neural networks to predict the sex of an individual simply based on their brain structure. And what we showed was that this neural network far outperformed other traditional machine learning approaches that are volume-based in predicting an individual's sex based on their brain structure. So that's another example of how we're applying bioinformatics to neuroscience. So to wrap up, I just want to emphasize again that bioinformatics is one of the few fields in biology where you can get a start with no specialized equipment or facilities. I hope those of you who are interested will do what it takes to build or take apart a computer, to install Linux, to go for these online tutorials, and then eventually when the time is right to seek formal training. Also, nearly all high-impact discoveries in biology now depend upon a bioinformatician, simply because the data sets are so enormous, and traditionally trained scientists don't have the skills needed to process and fully comprehend those data sets. Career prospects are currently good, but you really do need to go the extra mile to clearly differentiate yourself from other people who just have bioinformatics written on their CV and a little more than that. Okay, and then I want to acknowledge my graduate students whose work I showed here. And if you've got any questions, I'm happy to take them now, or I realize that I've kind of run out of time, but if you have specific questions, you can email me at that email address. You can also check out our lab website. So thank you guys for having me, and I'm happy to take any questions that might not have been asked.